The T critical for 95% confidence level was 1.67. Why for 95% confidence level? Because that was the minimum value in we used in order for us to accept our alternative hypothesis. Degrees of freedom during our experiment were 38. So, and the confidence level range that we found, so the level of our confidence was between 99.95% and 99.99%. So what's the conclusion? So what's the conclusion of our experiment according to all this data? So after performing the data analysis and performing the statistical analysis, we came up with the conclusion that our alternative hypothesis is supported, meaning that water vapor can diminish the rate of the cellular respiration in comparison with the in a similar way as the chemical toxins that are used currently by the farmers. Why we support that alternative hypothesis? Well, first of all, we saw that the mean specific rate mean of our experimental group was much less in comparison to our control, and our statistical analysis. So according to the rules where the T calculated is bigger than the T critical, we should accept our alternative hypothesis. If it's bigger for T critical, that represents 95% confidence level, and the precise range of our confidence was between 99.95 and 99.99%, which is more than enough for us to accept our alternative hypothesis. So that was the reason why we accept our alternative hypothesis, and we came up with the conclusion that water vapor indeed diminishes or decreases the rates of the cellular respiration. But where do we go from there? So what's our future studies? Well, the data that we were presenting to you today is just the preliminary data. It's just the beginning of our work. So first of all, what we want to see, in the future experiments that we want to do, today or last week, the experiment that we performed was the experiment where we just purely measured the rate of the cellular respiration exposed to the water vapor seeds. But we are not, although we believe that it will have a similar effect as the seeds that are exposed to the chemical toxins, we don't know for sure how significant the effect will be. So in the future studies, what we want to do, we want to take the same seeds and we want to expose them to the chemicals that are currently used by the farmers. And we want to measure the rates of the cellular respiration of these seeds after that and see the comparison. Are we going to achieve the same results in comparison to the seeds that are exposed to the water vapor or not? So that's one of the ways that we want to take our experiments further. Another way we want to take our experiments further, maybe we want to take it in this kind of direction. Yes, the farmers can sterilize the soil and plant their seeds which they want, but the question is this: where's the guarantee that the harmful plants or unwanted plants are not going to grow up? Maybe they will grow up at some point, and what will the farmers do at this point? So in order to answer that question, what we want to do in the future, probably we want to test both. We want to test some of the types of the unwanted plants by the farmers, and some of the types of the plants that are wanted by the farmers, the plants that farmers want to plant. And we want to expose them both to the water vapor and determine maybe there is an optimal point in which the unwanted or harmful plants get damages, and the plants that are wanted by the farmers stay untouched or stay safe. So this is just broadly one of the future directions where we want to take our studies. And this is the references that we used during our presentation, and with that I'll take some